Hello dear students, welcome to this program. Today we are discussing an interesting topic which is DNA topology, linking number and topoisomerases, organization of DNA in prokaryotes, eukaryotes and viruses, RNA structure, organelle DNA, mitochondrial and chloroplast DNA. Let me start with our first objective, DNA topology, linking number and topoisomerases. Cellular DNA is extremely compacted, implying a high degree of structural organization. The folding mechanism not only must pack the DNA, but also must permit access to the information in the DNA. This folding mechanism is accomplished by a process called supercoiling. Supercoiling means the coiling of a coil. A telephonic cord, for example, is typically a coiled wire. DNA is coiled in the form of a double helix, with both strands of the DNA coiling around an axis. The further coiling of that axis upon itself produces DNA supercoiling. DNA supercoiling is generally a manifestation of structural strain. When there is no net bending of the DNA axis upon itself, the DNA is said to be in a relaxed state. Supercoiling is an intrinsic property of DNA tertiary structure. It occurs in all cellular DNAs and is highly regulated by each cell. For circular DNA molecules, a topological property is one that is unaffected by deformations of the DNA strands as long as no breaks are introduced. Topological properties are changed only by breakage and rejoining of the backbone of one or both DNA strands. As DNA is a flexible structure, its exact molecular parameters are a function of both the surrounding ionic environment and the nature of the DNA binding proteins with which it is complexed. Because their ends are free, linear DNA molecules can freely rotate to accommodate changes in the number of times the two chains of the double helix twist about each other. But if the two ends are covalently linked to form a circular DNA molecule, and if there are no interruptions in the sugar phosphate backbones of the two strands, then the absolute number of times the chains can twist about each other cannot change. Such a covalently closed circular DNA is said to be topologically constrained. In order to fully understand DNA topology, there is need to familiarize with three key mathematical concepts which are twist, writhe and linking number. The linking number Lk for covalently closed circular DNA molecule is the number of times one strand crosses the other. For a relaxed molecule, the Lk can be referred to as Lk0. Lk linking number is a topological property of DNA. Lk can be expressed as the algebraic sum of two geometric properties, twist and writhe. Twist refers to the manner in which one strand winds around the second. Writhe refers to the non-planarity of the DNA axis. Lk linking number is equal to twist and writhe. Topoisomerases. Topoisomerases are the enzymes that catalyze the changes in the linking number of DNA. DNA supercoiling is precisely regulated process that influences many aspects of DNA metabolism. Every cell has enzymes with the sole function of unwinding and or relaxing DNA. The enzymes that increase or decrease the extent of DNA unwinding are topoisomerases. The property of DNA that they change is the linking number. These enzymes play an especially important role in processes such as replication and DNA packaging. There are two classes of topoisomerases. Number 1, type 1 topoisomerases, and number 2, type 2 topoisomerases. Type 1 topoisomerases act by transiently breaking one of the two DNA strands, passing the unbroken strand through the break and rejoining the broken ends. They change linking number Lk in increments of 1. A type 1 topoisomerase cuts one strand of a DNA double helix, relaxation occurs, and then the cut strand is reannealed. Cutting one strand allows the part of the molecule on one side of the cut to rotate around the uncut strand, thereby reducing stress from too much or too little twist in the helix. Such stress is introduced when the DNA is supercoiled or uncoiled to or from higher orders of coiling. Coming to type 2 topoisomerases. Type 2 topoisomerases break both DNA strands and change linking number Lk in increments of 2. A type 2 topoisomerase cuts both strands of one DNA double helix, passes another unbroken DNA helix through it, and then reanneals the cut strands. Examples of type 2A topoisomerases include eukaryotic topo2, E. coli gyrase, and E. coli topo4. Examples of type 2B topoisomerase include topo6. Type 2 topoisomerases utilize ATP hydrolysis. Our second objective is organization of DNA in prokaryotes, viruses, and eukaryotes. Organization of DNA in bacteria. A single E. coli cell contains almost 100 times as much DNA as a bacteriophage particle. The chromosome of an E. coli cell is a single double-stranded circular DNA molecule. It's 46,39,675 base pairs long and have a contour length of about 1.7 mm, some 850 times the length of the E. coli. In addition to the very large circular DNA chromosome in their nucleoid, many bacteria contain one or more small circular DNA molecules that are free in the cytosol. These extra chromosomal elements are called plasmids. Most plasmids are only a few thousand base pairs long, but some contain more than 10,000 base pairs. They carry genetic information and undergo replication to yield daughter plasmids, which pass into the daughter cells at cell division. Plasmids have been found in yeast and other fungi as well as in bacteria. In many cases, plasmids confer no obvious advantage on their host, and their sole function seems to be self-propagation. Organization of DNA in viruses. Viruses are not free living organisms, rather they are infectious parasites that use the resources of a host cell to carry out many of the processes they require to propagate. Many viral particles consist of no more than a genome, usually a single RNA or DNA molecule surrounded by a protein coat. Almost all plant viruses and some bacterial and animal viruses have RNA genomes. These genomes tend to be particularly small. For example, the genomes of mammalian retroviruses such as HIV are about 9,000 nucleotides long, and the genome of the bacteriophage Q has 4,220 nucleotides. Both types of virus have single-stranded RNA genomes. The genomes of DNA viruses vary greatly in size. Many viral DNAs are circular for at least part of their life cycle. During viral replication within a host cell, specific types of viral DNA called replicative forms may appear. For example, many linear DNAs become circular and all single-stranded DNAs become double-stranded. A typical medium-sized DNA virus is bacteriophage lambda, which infects E. coli. In its replicative form inside cells, DNA is a circular double helix. This double-stranded DNA contains 48,502 base pairs and has a contour length of 17.5 meters. Bacteriophage X174 is an example of smaller DNA virus. Although viral genomes are small, the contour lengths of their DNAs are typically hundreds of times longer than the long dimensions of the viral particles that contain them. Coming to the organization of DNA in eukaryotes. A yeast cell, one of the simplest eukaryotes, has 2.6 times more DNA in its genome than an E. coli cell. Cells of Drosophila, the fruit fly, used in classical genetic studies, contain more than 35 times as much DNA as E. coli cells, and human cells have almost 700 times as much. The cells of many plants and amphibians contain even more. The genetic material of eukaryotic cells is apportioned into chromosomes, the diploid 2n number, depending on the species. A human somatic cell, for example, has 46 chromosomes. Each chromosome of a eukaryotic cell contains a single, very large duplex DNA molecule. The DNA molecules in the 24 different types of human chromosomes, 22 matching pairs plus the X and Y sex chromosomes, vary in length over a 25-fold range. Each type of chromosome in eukaryotes carries a characteristic set of genes. The DNA of one human genome, 22 chromosomes plus X and Y or 2X chromosomes placed end to end, would extend for about a meter. Most human cells are diploid and each cell contains a total of 2 meters of DNA. An adult human body contains approximately 10 raised to the power 14 cells and thus a total DNA length of 2 into 10 raised to the power 11 kilometers. Compare this with the circumference of Earth, which is 4 into 10 raised to the power 4 kilometers, or the distance between the Earth and the Sun, which is 1.5 into 10 raised to the power 8 kilometers, a dramatic illustration of the extraordinary degree of DNA compaction in our cells. RNA structure. RNA structure is simpler than DNA structure. RNA differs from DNA in 3 respects. First, the backbone of RNA contains ribose rather than 2-deoxyribose. That is, ribose has a hydroxyl group at the 2' position. Second is, RNA contains uracil in place of thymine in DNA. Uracil has the same single ring structure as thymine except that it lacks the 5' methyl group. Third is RNA is usually found as a single polynucleotide chain. Except for the case of certain viruses, RNA is not the genetic material and does not need to be capable of serving as a template for its own replication. Rather, RNA functions as the intermediate, the mRNA, between the gene and the protein synthesizing machinery. Another function of RNA is an adapter, the tRNA, between the codons in mRNA and amino acids. RNA can also play a structural role as in the case of RNA components of the ribosome. Yet another role for RNA is as a regulatory molecule which through sequence complementarity binds to and interferes with the translation of certain mRNAs. Finally, some RNAs, including one of the structural RNAs of the ribosome, are enzymes that catalyze essential reactions in the cell. In all of these cases, the RNA is copied as a single strand of only one of two strands of the DNA template, and its complementary strand does not exist. RNA is capable of forming long double helices, but these are unusual in nature. Now our last objective, which is organelle DNA, mitochondrial and chloroplast DNA. Eukaryotic cells also have organelles, mitochondria and chloroplasts that contain DNA. Mitochondrial DNA molecules are much smaller than the nuclear chromosomes. Each mitochondrion typically has 2 to 10 copies of this mitochondrial DNA molecule, and the number can rise to hundreds in certain cells of an embryo that is undergoing cell differentiation. Plant cell mitochondrial DNA ranges in size from 2 lakh to 25 lakh base pairs. Chloroplast DNA also exists as circular duplexes and ranges in size from 1,20,000 to 1,60,000 base pairs. Mitochondria and chloroplasts divide when the cell divides. Their DNA is replicated before and during division, and the daughter DNA molecules pass into the daughter organelles. Cellular respiration, that is the oxidation of nutrients to generate energy in the form of adenosine 5' triphosphate ATP, takes place in the mitochondria. These organelles are roughly 1 to 2 micrometers in diameter, and there may be 1000 to 2000 per cell. They have a smooth outer membrane and a convoluted inner membrane that forms protrusions called cristae. They contain a small circular DNA molecule, mitochondrial specific RNA, and ribosomes, on which some mitochondrial proteins are synthesized. However, the majority of mitochondrial and chloroplast proteins are encoded by nuclear DNA and synthesized in the cytoplasm. These later proteins have specific signal sequences that target them to the mitochondria. Mitochondrial DNA is replicated via RNA Okazaki fragments. Mammalian mitochondria contain 2 to 10 copies of their 16 kb circular chromosome. The Paul gamma mediated replication of this chromosome occurs unidirectionally from a single origin. In this process, as Einhold showed, the lagging strand Okazaki fragments are entirely synthesized as RNA. The RNA is then replaced by DNA, although the way this occurs is poorly understood. One possibility is that this RNA is excised in much the same way as the primers in the nucleus, that is through the actions of RNase H1 and FAN1, followed by the synthesis of lagging strand DNA by pol gamma. The chloroplasts of plants are the site of photosynthesis. The light-dependent assimilation of carbon dioxide and water to form carbohydrates and oxygen. Though larger than mitochondria, they have a similar structure except that in place of cristae, they have a third membrane system, the thylakoids, in the inner membrane space. These contain chlorophyll, which traps the light energy for photosynthesis. Chloroplasts are also partly genetically independent of the nucleus. Both mitochondria and chloroplasts are believed to have evolved from prokaryotes, which had formed a symbiotic relationship with a primitive nucleated eukaryote. Here we come to the conclusion of our today's topic. Hope you understood it well. Have a nice time.